biology students, and welcome to my classroom. Today we're going to be doing a lab to see if we can get some evidence for how aquatic plants can cycle carbon. So today we're going to be using an aquatic plant called Elodea, which I have in my little beaker here. And we're going to be using an indicator called BTB, which stands for bromothymol blue. So I have 2 beakers of water here. Okay, both of these are water just straight out of the sink. And what I'm going to do, this one, I'm going to go ahead and put 10 milliliters into each of my test tubes. So I'm going to go ahead and put 10. Now water is neutral, so it's at a pH of 7. So BTB is an indicator used to show that a solution is acidic. So if an acid is in there, then it will change color. So I'm going to get some of my BTB here, and I'm going to add about 10 drops ish into my test tube. So I'm going to get a nice blue color. So I'm going to mix that up. So blue. So showing that this is a neutral solution. Okay, so I'm going to shake again. Okay, we got 2 of these that are going to be blue. Now these 2, I actually want them to be yellow. So what I'm going to do is, the way to make them yellow, is to add carbon dioxide. The carbon dioxide mixes into the water and forms what we call carbonic acid. So where can I get some carbon dioxide? I know. I breathe out carbon dioxide all the time, especially since I talk so much. So I'm going to get a straw and I'm going to blow into this beaker full of water. So I'm adding carbon dioxide into my water. Okay, so now my BTB should indicate the presence of carbonic acid in my water. So I'm going to go ahead and pour 10 milliliters in. Okay, 10 milliliters in here. There we go. And now I'm going to add BTB, and if there's enough carbon dioxide to form carbonic acid, then it should turn yellow. So, okay. So started off a little bit green, shake it up, I got myself a nice yellow solution. Let me mix that up really good. Okay, so I'm going to add some more to this one. There we go. Give it a nice shake. Looks a little like the urine of somebody who doesn't drink enough water. So we got a nice yellow solution. All right, so remember the formula for photosynthesis is plants take in 6 carbon dioxides plus 6 waters plus energy from the sun, and that gives them the ability to make glucose and then release oxygen. The formula for cellular respiration, plants get the glucose that they've made, bring in oxygen, and they produce carbon dioxide, water, and energy in the form of ATP. So for this one that is neutral right now, I'm going to take one of my sprigs of Elodea and I'm going to put it in there. Okay, and I am going to put a lid on it. Oops, hold on, let me get my lid. Okay, push it down nice and tight. Get it to snap on there. And then I'm going to wrap it in aluminum foil so that no light can get in here. So what do you think is going to happen in the next 2 days? What color do you think the solution is going to turn? Okay. What do you think is happening in this tube? Now in this other blue tube, this is my control. So the only thing that's going to be different between this tube and my experimental tube is the presence of the Elodea. So I'm going to snap that lid down on there nice and tight, grab my tin foil, and I'm going to wrap it up so no light gets in there. What do you think is going to happen to the control? Okay, it has the same things in it except for the Elodea. What color do you think it's going to be in 2 days? Okay, over here, I have a tube that I have infused carbon dioxide into, so we have a nice yellow color. So I'm going to take my other sprig of Elodea, and I'm going to put it in there. Use my straw to push it down. Put the lid on it. This one is going to sit under the light. Okay, so I'm not going to put anything on the outside. It's just going to be Elodea hanging out in the light. And then this is my control tube, no Elodea. It has the same solutions in it. What do you think is going to happen to the control tube? All right, so now we're going to let these guys sit under the light for 2 days, and then we're going to come back and see what happens. Hi students, welcome back to the classroom. So I have let these tubes sit under the light and now we're trying to see what happened. So starting with our two tubes over here that we left uncovered, okay, here was our control. Remember our control started off as yellow, all right? And then here's our tube that has Elodea in it. It's been sitting under the light. So you'll notice that it definitely had a color change. So let me see if I can get a, there we go. So you can see it went from yellow, it started off yellow and now it's blue. So what happened in this tube that allowed it to change colors? And why didn't the control change? Good questions, good questions. So over here, I'm going to unwrap these two tubes that have been in the dark. And let's see what happened there. Unwrapping, unwrapping. Oh, okay. So I got a yellow tube, and let's see what's going on in this one. Hit it really up. So my control tube started off blue and is still blue, but my experimental tube started off blue and now it is yellow. So what happened in this tube that caused it to turn yellow? Good questions. So let's go ahead and analyze our data and see what we come up with.